So if we see the last example, we have taken the yellow P or green P. So we are referring about the character of the P. So whether it's a color, right? So we are referring about the color of the P. So we are looking into the only one character in that. So in that one character, we have a two contrasting alleles or two contrasting traits, whether it's a green, whether it's a yellow. So that's why as we are looking into the one character, so we call that as a monohybrid cross. Understood? So in monohybrid, mono means we are looking into one character that is color of the P, whether it's a yellow or green, the contrasting traits. So we studied about only one character and we did the cross with only to experiment about the one character. So it's called as a monohybrid cross. Whereas here we are saying it's a dihybrid means, so within our experiment, we will be studying about the two characters. Okay. So in this cross, we'll be looking into, or we will be drawing the inference over two characters. So that's why it is called as dihybrid cross. Here we are hybriding between the one character, green and yellow. Here we are hybriding with the two different characters between the two plants. In monohybrid cross, we are taking one character and whatever the contrasting plants are there, the green and yellow, we are hybridizing both of them. So that's why it is called as the monohybrid cross. Whereas here we are taking two different characters and we'll be drawing the inference between the two different characters. So this cross is called as the dihybrid cross. So here it's a cross between two individuals with two different observable characters. In the monohybrid cross, plant user differed in one character. Okay, so that's a pod color. So whether it's a yellow or green. So what Mendel has done is he also checked whether the whatever the laws he stated are applying to the dihybrid cross or not. Okay, so whether when the plant is having several different characters, are they united when they hybrid or not? That is what he is studying. Understood? So when a plant is having different characters, and when he is doing hybridization, that means he is taking the dominant and recessive characters. And when he is doing this hybridization, so are they, whatever the laws he stated, are they applying to it or not? Okay, when he is drawing inference about the different characters, all these laws which he stated, are they applicable or not? That is what he studied under the experiment of dihybrid cross. So you can see here, we are taking the pea plant with a round shape and yellow seeds. That is one character. So here we are taking round and yellow color. And the other side, we are taking green seeds which are wrinkled. So this is a recessive character. It is a wrinkled shape and green color. So wrinkled and green. So these are the recessive characters. Whereas on the other side, it's a round and yellow, which is a dominant character. So he has taken two different plants. One is the dominant plant, which contains the round seeds and yellow. The other one, which contains the wrinkled and green seeds. So he is doing the cross between the two. Okay. And he obtained the F1 generation, which is, there are different. Okay. So all are like round and yellow seeds. So if you see here, the round and yellow dominant character can be represented like this, YY and RR. Whereas here it is YY, RR. Okay. So whereas here R alleles refer for the wrinkled, whereas capital R refers for the round, and capital Y refers for the yellow, whereas small y refer to the green. Okay. And we know that Mendel always, when he is taking the parent, he always take the pure lines. So pure lines, or he will take the true breeds. Okay. So these pure lines, one of the pure lines are the dominant character, the other pure lines are recessive character. Here it is a round yellow, so which is capital Y, capital Y, capital R, capital R. The other one is small y, small y, small r, small r. You understood this? So what he has done in dihybrid cross, what parents he has taken?
Sir, could you explain again?
Okay. See, Mendel always want to take the pure lines or true breeds. Gauri, you didn't there for the last class. So do you know what are pure lines and what are true breeds means?
Yes, sir.
If not, I'll explain this. Okay, fine. So here he is taking the pure lines of the dominant character. What are the dominant characters here? Is one is the round, the other one is yellow seeds. So round and yellow seeds, if it is round is a dominant and homologous, that means it is capital R, capital R. And if it is yellow, that means capital Y, capital Y. And he is taking both the characters at one time. So I'm writing this YYRR. This is one parent in the dihybrid cross. On the other side, he is taking another parent or another plant, which is wrinkle seeds and green seeds. So it is smaller, smaller, and small y. So together, I am writing it as small y, smaller, smaller. So this is parent B, this is parent A, or you can say plant A and plant B. So this is the dominant plant, and this is the recessive plant. And he's taking these two, dominant plant and recessive plant, and he is doing the cross breeding between them. And I explained how does we do the cross breeding. So first we'll convert whatever the bisexual plant into the female plant, and we'll take the anther and we will crossbreed between the two. So we are not doing self-breeding here, we are doing the crossbreeding here artificially. So after doing the crossbreeding, we will get the seeds and we'll sow them in the soil. And whatever the plants that are obtained after sowing the seeds, those are all F1 plants. Or we can say the F1 generation or filial 1 generation. So in the filial 1 generation, he saw that all the plants were round and yellow seeds. So whatever the plants he got here, all are round and yellow. That means they are showing only the dominant character. They didn't show any recessive character in F1 generation. That's the same thing we observed in the monohybrid cross as well. Yes or no? So we saw all are yellow P. We didn't see any green P in F1 generation. The same way here also, we are seeing all are round and yellow. That's a dominant trait. And we didn't see any wrinkled or green in F1 generation. So whatever the monohybrid cross, whatever the inference we draw, the same inference is showing here. The same observation we are seeing here. Is that, guys, you understood? So for whatever I said.
Yes, sir.
Yes.
Okay. Please, if anyone have a doubt, please ask me. I'll explain it again. Okay, someone tell me what does the Mendel do in F1 generation after F1 generation? So before F1 generation, he took the two parents. One is a dominant parent, one is a dominant plant, the other one is recessive plant, and he did the cross breeding, and he obtained the F1 generation. And what does the Mendel do in F1 generation? After F1 generation,
He'll crossbreed the same thing.
just consider your words again and tell it again. He
Self
crossbreed.
I mean, okay, self-pollinate.
Yeah, that is very important. He didn't do the crossbreeding. He self-fertilized the F1 generations. So remember, guys, in F1 generation, he didn't do crossbreeding. It did self-fertilization.
No, I meant crossbreeding meaning the same meaning.
They are different. Self-fertilization is different. Cross-fertilization is different. Cross-fertilization, where you are taking two different types of plants and you are doing it, which are genetically different. Whereas here, self-fertilization means you are taking the same genetical parent and you are doing it again. You are doing with that plant again. So guys, don't confuse with the self-fertilization and cross-fertilization. We have seen cross-fertilization is a different, self-fertilization is different. It's autogamy, whereas the other one is not autogamy. Okay, so both are not same. So yeah, he then self-fertilized the F1 generation. So we know that in monohybrid cross, it is capital R smaller. Whereas in the dihybrid cross, it is capital R smaller, capital Y small y. This is the dihybrid F1 generation parents. Okay, so from that, he did again the crossbreeding with the same, sorry, he did the self-fertilization here in F1 generation. And when you see the F2 generation, he got 4 different kinds of plants. So what are the 4 different kinds of plants is? One, it is yellow and round seeds. He got a yellow and wrinkle seeds. He got green and round seeds. And fourth, he got green and wrinkle seeds. Okay, so these are the four different kinds of plants he got in the F2 generation. So one is yellow and round seeds, two yellow and wrinkle seeds, three green and round seeds, four green and wrinkle seeds. If you see in the mono hybrid cross, we only got two different kinds of plants. One is the yellow and the other one is green. But here we got a four different kinds of uh, characters in the F2 generation. One is yellow and round, yellow and wrinkle, green and round, green and wrinkled. Understood? What is the slight difference between the F1 and F2 generation? So you can see here. So there are round and yellow, round and yellow, round yellow, round yellow, wrinkled yellow, round yellow, wrinkled yellow. So round green, wrinkled green. Okay, and wrinkled yellow. So these are the F2 generation thing. And what is this arrangement? We call it as. So if we take the parents out and we draw this inference, so what is this type of graphical representation?
Punnett square.
Yeah, it is called as the Punnett. Okay, so remember in the Punnett square, we will be writing the parental gametes, okay, outside the box. So remember, these are all parental gametes. We will be writing outside the box. And inside, we'll be writing the phenotype and genotype of the F2 generation. So this is the phenotype and this is the genotype. Okay, so phenotype and genotype, phenotype, genotype, phenotype, genotype. Understood? So this graphical representation, we call it as the Punnett square. And if you see the observation here, how many round and yellow are there? So 1, 2, 3, 4, 5, 6, 7, 8, 9. So there are total 9 are present. Yes. And how many wrinkled yellow are there? 3. So 1, 2, 3. So there are 3 wrinkled yellow. And how many round green are there? 1, 2, 3. So 3. And how many wrinkled green is there? Only 1. So this is the phenotypic ratio of the F2 generation. So remember this. So the phenotypic ratio of the F2 generation is 9:3:3:1. And if you draw the genotypic ratio, it will go so much a long length. So you can see here. This one. 1:2:1, 2:4:2, 1:2:1. So if you guys ask, sir, should I remember this ratio and this entire sequence? Yeah, it will be very helpful if you remember this. How it will be helpful is they can ask, they can give like any character and they can ask, so how many capital Y, smaller obtained? Then you can't draw the entire Punnett square in 1 minute and you tell that. So instead you can remember this ratio and you can say it easily. Then how to remember this? You can remember this way. You can write in a 3 sets. Okay. So set number 1, Y, R, R. Y, capital R, small r. Y, smaller. This is the set number 1. And set number 2. So Y, capital R, Y, capital R, smaller. Y, smaller. So in set number 1, I have taken yellow, the all are homologous dominant. In the set number 3, I have taken all are recessive homologous, yellow. And I wrote capital R, smaller, capital R, smaller. So this is the set number 1. This is the set number 2, set number 3. And we have a middle set, that's a set number 2. So that you can remember like this. You will be taking the heterozygous, capital Y, small y, capital Y, small y, capital Y, small y. And the same thing you will be following, capital R, smaller. You understood? So how did I write this? So first I have taken the set 1. So in which I have taken the homologous yellow. And then I have taken the homologous green as my recessive character. See, here I am taking yellow and green, but wherever, if he is given any dihybrid cross, you write first you take one character and you start writing it. Okay, so first you take a one character and you start writing this. So first I am taking here the yellow character, the dominant homologous, and then I am taking the recessive character, and then I am taking the heterologous, the dominant character. After that, I am whatever the other set is there, or the other character is there, I'm taking it like this: capital, smaller, smaller, smaller. The same for all the three sets. Is that clear, guys? So what I'm trying to hear, how I wrote this. This is very helpful. It will come so handy for the exam, for board as well. If they give any dihybrid cross, here they have given yellow round. They can give any type. They can give the seeds, or they can give anything like the height of the plant. Instead of yellow, he can give the height of the plant and the shape of the seeds. So you should be able to write the genotype without drawing the entire Punnett square. So tell me if you understood up to here. Just say yes or comment in the box yes.
Yes, sir.
Yes, sir. Understood.
Yes, sir.
Okay.
Yes,
So
sir.
now how I'll give the ratio numbering is, so for set 1, I'll be just writing 1:2:1. That's a monohybrid cross we know. So the same ratio you will be giving for here as well, 1:2:1. For set B, as we are taking the two dihybrid, so you multiply this ratio into 2. So that will be coming as the set B values. So which is 2:4:2. And that's it. You got the values of the genotype. So you understood how we are drawing this.
Yes, sir.
So this is the easy method to remember. So take the first dominant trait, capital Y, capital Y, capital Y, and take the recessive, small y, small y, small y. And then whatever the second character is there, you will be giving capital, smaller, 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 capital, smaller, smaller, smaller. And for the middle set, you will be giving this way. In middle, you will be taking the heterologous. So capital Y, small y, capital Y, small y, capital Y, small y. And the ratios will be for set 1, 1:2:1. For the set C, 1:2:1. For the middle set, you are giving as 2:4:2. So from that, you can predict this genotypic ratio of the dihybrid cross. Is that clear?
Yes,
So
sir.
from this, you can easily tell. If someone says, give me the heterologous, how many heterologous yellow round are there? So what's the answer here? How many seeds after the dihybrid F2 generation, heterologous, yellow and round are there? How
One.
many are there? Heterologous or heterozygous, yellow, round. How many are there in the F2 generation? So what is meaning of heterologous, yellow and round? It is heterologous yellow. That means capital Y, small y. Heterologous, heterozygous round. That means capital R, smaller. How many are there? This one.
One, sir.
Oh.
Guys, how many are there? Is it only one?
Oh,
4.
Oh, okay.
Now tell me, in F2 generation, how many heterologous yellow and homologous round are there? So now the question is heterologous.
sir.
Two.
Hmm.
You asked heterozygous yellow round. So then in the first one also comes heterozygous round, right?
First one where?
Hetero means two, right?
Yeah, no, I mean capital Y, small y, capital R is also round only, right?
No, but it
I
just means
said
that
for
like,
the phenotype, not for the. See, capital Y, capital R smaller. Where is that?
No, sir. I mean the second one. You didn't mention dominant or recessive. So both are yellow only, right?
See, my question is this: heterozygous, yellow, round. Heterozygous yellow means capital Y, small y. Heterozygous round means capital R, smaller. So why you will be taking capital Y or capital R, capital R? These are homozygous, right?
No, no, I meant capital Y, small y, and capital R, capital R. That
Then it
kind of.
is not heterozygous round. It is homozygous round.
Okay.
You understood? See,
Okay.
here heterozygous means I'm mentioning for both yellow and round.
Okay, yeah, that
Okay.
I get it.
Heterozygous yellow, heterozygous round. Is that okay?
Yeah, like that. Yeah, got it.
Yeah. So now Tanzila, you answer this. Heterozygous yellow and homozygous wrinkled. So tell me, how many are there here? So here heterozygous yellow means capital Y, small y, and homozygous wrinkle means small y, smaller. So how many are there here? 2 are there. Nice. So F1 generation, we did the cross like this. This is the F1 generation, we crossed. We did the self cross, and this is the F2 generation we obtained as the results. So yeah, so what is the dihybrid cross inference that is Mendel drawn is? One, so the round seed is the dominant trait, wrinkle seed, it's a recessive trait. And round and yellow seeds hence form the dominant trait, whereas green and wrinkle seeds form the recessive phenotype. Okay. And whatever that you are seeing, like the round green, like the hybrid, so round green, or the round yellow, so which are also formed as the intermediate propositions. Okay. And one more thing, you remember this. So all these will indicate there is a different characters are passed out to the gametes independently. So suppose you are taking the round yellow, that means capital R, and capital Y, and you are getting smaller, small y. But if you see whatever the F2 offsprings that we got, F2 plants we got, some of them are round green. That means it is round. And for suppose if you see here, this is the dominant trait and this is the recessive trait. Okay. So you can say from this, you can say, so whenever the genes are forming the gametes, so whatever the two characters are there, they are independently moving to the gametes. They are not dependently. For suppose the dominant yellow is went, so that gamete also should get the dominant seed shape. It's not like that. They are moving independently. Understood? So alleles of different characters, they are passed on to the gametes very independently. Okay, they are not depending on each other. Here the round is not depend on the color of the seed, or here the wrinkle is not depend on the color of the seed. Is that clear? That's one of the major important here. So whenever there are two characters we are taking, so each of the character is independent to the other when they are making the gametes. Okay, so it will come like this. For suppose when it make the gamete, it will come like this. One is capital R, capital R, capital Y, smaller, small y. So out of this, so you are getting total four different types of gametes. In that, which of the gamete will intermix, that will depend on the offspring that is formed. For suppose, let's say this is A, B, C, D, and this is 1, 2, 3, 4. So this is the parent A, this is the parent B. So which of the gamete will fertilize the other one? So it depends on those characters we'll get. So whether we are getting the round and whether it is like this, capital R, smaller, and this capital Y, small y, or we can get like if the parent is having small y, like this. So we can also get this small y and small y. So you understood what I'm saying here, whatever I'm trying to say here. So when there are two characters, they are independently passing to the gametes. So round is not influencing the yellow is not influencing the green or whatever. Okay. So they are passing out independently. This is the most important thing you need to understand in the dihybrid cross. So whatever the characters we are taking, they are independent to each other when they are forming the gametes. So you can see here. So total, we can get a 4 different kinds of gamete: capital Y, capital R, y, smaller. So from that, he stated this law. So law of independent assortment. Okay. So based on whatever the dihybrid cross that Mendel has observed, he stated the new law that's called as law of independent assortment. What does it says? When two pairs of traits are combined in a hybrid, that means two different types of traits. That means one we are taking. Okay. So segregation of one pair of character is independent of the other pair of the character. That means whenever they are making the gametes, these two will not be combined. They will be separated. Okay, independently. So that is what the law of independent assortment says. So when two pairs of traits are combined in a hybrid, segregation of one pair of character is independent of the other. First, suppose you take this one, like capital Y, capital R, small r. Let's take the F1 generation. So how they are forming into the gametes? First, we can get capital Y, capital R, like these two, or we can get capital Y, small r, or we can get small y, capital R, or we can get small y, small r. The same way happens here as well. Capital Y, capital R, capital Y, small r, small y, small capital R, small y, small r. So these are the 4 different types of gametes we can form. And which one is fusing with which one? So that will depends. Okay. For suppose this capital Y, capital R can fuse with this or this or this or this. So like that. So there are total 4 and each 4 will form the other 4. So we'll be getting almost the 16 characters. So 4 is to 3 is to 3 is to. Wait. How many? Yeah. We'll be getting 16. Actually, we are simplifying this ratio. So we'll be getting total 16. Okay. So if you see the Punnett square, actually it's 9 is to 3 is to 3 is to 1. Sorry, my bad. So if you see how many are there, so total 16 are formed. You understood? First, suppose I'm taking this A. This A can combine with any of the 4. So with A, I can get 4 different types. And take the B. This B can combine with any of the 4. And the B also gives total 4. So the same with the C and D. So total from each of them, I am getting together as 16. So this is what the law of independent assortment says. If you guys have any doubt, please ask me. If you are understood, please say yes. So what is meaning of law of independent assortment?
Understood, sir.
Understood.
Okay. So this is how it is forming the Dihybrid cross. Okay. So let's see few things in the Dihybrid cross. So first one, consider the segregation of one pair of genes, capital R and small r. 50% of the gametes have the gene capital R and the 50% have the small r. So you take this and you segregate this. So we'll be getting 50% this and 50% this. Okay. Each gamete having either capital R or small r allele should also have the allele capital Y or small y. Okay. So whenever they are making the gametes, they can have to make either with the capital R and capital Y, or they can also make capital R, small y, or they can make small r, capital Y, or small r, small y. So that's what he says here. Each gamete having either capital R or small r, they should also contain capital Y or small y. Okay. And the segregation of 50% of R and 50% of R is independent of the segregation of the Y and small y. So whenever, first suppose you take capital R, small r, capital Y, small y. So whenever these two are segregating, that means when they are separating, it is independent of these two. So when A is splitting out into capital R and small r, the B is also splitting into capital Y, small y, but A and B are independent to each other. That means as A is splitting into capital R, it is not influencing the Y. Okay. So one character is not influencing the other character. That's what the previous one says. That's a law of independent assortment says. So one is independent over the other character when they are segregating. Next one. Thus, there are total 4 genotypes of gametes are there. Okay. So what are those 4? You can see here. These. So we got the total 4 genotypes of gametes. And 4 types of pollen and 4 types of egg. That's a 4 types of male and 4 types of female. And which one will mix to which one, that depends on the type of species we'll get, the type of organism we'll get. For suppose capital R, capital Y, capital R, small y, small y, capital Y, small r, small y. So which one is mixing with capital R, capital Y, capital R, small y, small r, small r, capital Y, and small r, small y. So these are from the pollen and these are from the egg. So which one is mixing with which one? So that will tell the which seed it will form. For suppose this will mix with this, then we'll be getting the seed like this. If this is mixing with this, we'll be getting seed like this. So which one is mixing with which one? So we'll get that particular type of characteristic organism. Okay. So total, the 4 types of capital R, capital Y, capital, these, each frequency is 25% or 1/4 of the total gamete produced. Okay. So whatever it is, 25% out of the 100 or 1 for every 4. Understood? So this is having 25% chance, this is having 25% chance, this is having 25% chance, and this is having 25% chance. So based on that, this is the frequency. So out of 100, each one will be having 1/4 of the proportion or 25% of the proportion out of 100. Understood?
Yes, sir.
So tell me this. So if you got the answer, please directly inbox me. Don't allow it. Just inbox me directly. So whoever feels whatever the answer. And it's okay. You try or it's okay. You don't try. No problem. So reminding all, just give me the answer. Directly inbox me. Don't send for all. Just send it to me. Only to me. See what you said here. The question. A cross was performed between the tall plant, which is a dominant heterozygous trait. So is telling about the characteristic of this tall plant. So the tall plant here is, it is a dominant, but it is heterozygous trait. That means it's not having homologous. They are not identical alleles or traits are present here. They are heterozygous. That means it is a tall plant. So I can write capital T or capital T small t. But he's saying heterozygous trait. So I'm not taking this. I'm taking this. Capital T small t. Okay. And with green seeds. So that means he's saying small y, small y. And what is the other thing he's saying? Dwarf plant. That means it is small t, because we don't have other option for dwarf. It's only capital small t with a heterozygous yellow seeds. That means it is capital Y, small y. So he did the crossbreeding between these two. And what he is asking here, what is the phenotypic ratio of offsprings that are dwarf and yellow? So this we cannot tell directly with the using the ratio. We need to go with the calculation, drawing the Punnett square. Listen this is very important. So first what I'm doing, so whatever the parent I have taken, now I'm drawing the gametes. So first what I did like this, capital T, small y. So that's my first gamete. Now capital T, small y. That's my second gamete. Third gamete is small t, small y. Fourth gamete is small t, small y. So I got gametes for this. Do the same for here as well. Small t, capital Y. Small t, small y. The same way here. Small t, capital Y. Small t, small y. So these are the gametes I got. Okay. So by using these gametes, now I'm very quickly drawing this Punnett square. So this will be my dwarf and yellow. 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 Is there anything else? Dwarf and yellow. This will not come. This will not come. Okay. So total we have 4. So the phenotypic ratio of dwarf and yellow plants is, understood this. See how he asked this, he's asking the proportion out of all. So out of all, that means out of the total plants, how many plants are dwarf and yellow? You understood? So what is the phenotypic ratio of offsprings that are dwarf and yellow out of total plants? So total plants are 16. Do you remember I told you the proportion initially? So 1/4. You remember that? Like out of, in the monohybrid, I told you, out of 4 plants that we get, so how many homologous dominant? So we'll take 1/4. How many are heterologous dominant, we'll take 2 by 4. How many are recessive? That means we'll take 1 by 4. From that, we got 1:2:1 ratio. Do you remember this? I think in the yesterday evening class, I said this. Nitya, Anusha, do you remember this?
Yes,
That
sir.
means we are
Yes, sir.
comparing the proportion. Proportion is the number of plants of having a particular character by total number of plants. So number of plants having particular character by total number of plants. So in this case, how many dwarf and yellow are there? We got the 4. And how many total plants are there? 16 are there. So the ratio will be 1 by 4. So the answer is 6. The question is correct only. Okay. So whenever he says a particular plant, that means you will need to compare this number of plants with the total number of plants. Is that clear for everyone? How we got the answer 1:4?
Yes, sir.
Shall I repeat or you understood it fully?
I understood it fully, sir.
What about the others? Gauri,
Understood.
Nitya, Anusha?
Understood, sir.
Understood,
sir.
Okay, okay. Okay, so let's see some of the important formulas. So type of gametes, 2 power n for all gametes. First, suppose if I am taking only one character, that means I'll be getting only 2 different types of gametes. Okay, so here n indicates the number of characters we are taking or number of heterozygous gene pairs are present. Okay, first suppose if we are taking only one character, that means capital T small t. But if we are taking 2 characters, that means these. So at that condition, n will become 2 for dihybrid cross. For monohybrid cross, it will be 1. Okay, so remember this. So in order to find out what are the number of phenotypes, it is 2 power n. First, suppose for monohybrid cross, we are taking only one character or only one heterozygous. That's a 2 power 1, which is 2. So we know that that's a yellow and green for monohybrid. If you are taking dihybrid, that means 2 power 2, which is nothing but 4. So we saw that what are the 4 different types? Round and yellow, round and wrinkled, green and yellow, green and wrinkled. So there are total 4 different types of phenotypes we have. And types of genotype, 3 power n. So we know it's a 3 power 1, that is 3. So that's how we got capital, smaller, smaller, smaller. This is for monohybrid. For dihybrid, we have seen 3 square. That means 3 into 3, which is 9. And we saw the ratio 1:2:1 and 2:4. What is that? 2:4:2, 1:2:1. So total how many are there? 1, 2, 3, 4, 5, 6, 7, 8, 9. So we got total 9 genotypes when we are taking 2 different types of characters or dihybrid. You understood this? How we are using the formula? If he is giving like 3 different characters and if he asks how many probability of the phenotypes will be formed, you will be taking 2 power 3. And genotypes will be 3 power 3. And types of gametes will be formed is 2 power 3. Right now, this for you, it's only we have only dihybrid cross. We are not going for trihybrid or multi-hybrids. Okay, is that clear? So they can ask questions like this. So if there are total 3 different types of 3 different types of heterozygous genes or 3 different types of characters, so what will be the number of phenotypes are formed? What will be the number of genotypes formed? You can take like 3 power n, 2 power n, and how many gametes will be formed? 2 power n. Understood? So how we use this formula?
Yes,
Is this
sir.
clear?
Yes, sir.
Yes.
Yeah. So you can see here. So types of gametes for monohybrid cross. So we can see here, there are total 2 gametes will be formed, either capital V or small v. Okay. And based on that, how many phenotypes will be formed? What is the formula for phenotypes? 2 power n, that is 2 power 1, which is only 2. So you can see here, only 2 types of phenotypes are there. Okay. And if you ask about the genotypes, how many genotypes are there? So 3 power n, that's a 3 power 1, which is nothing but 3. So you can see here, capital V, capital V, capital V, small v, small v, small v. So there are total 3 types of there. So the answer is 3. So the same way for dihybrid cross, 2 power n. So you will be taking 2 power 2, which is 4. So you can see here. So total 4 gametes are formed. Capital Y capital Y smaller, small y capital, small y smaller. And if you ask about the phenotypes, it's a 2 power 2, which is 4. So you can see here, round yellow, wrinkled yellow, round green, wrinkled green. So you can see here. And what's the genotypic ratio? It's a 3 power n, which is nothing but 3 power 2, which is nothing but 9. So you can see here, total 9. And it's in the ratio 1:2:1 is to 2:4:2, 1:2:1. So from this, you can easily tell how many of them are formed so and so. So tell me this. Use the probability method to calculate the number of gametes and genotypes respectively in a self-cross of capital A small a, capital B small b, capital C small c. So how many are present here? How many heterozygous are present here? Or how many characters are present here? There are total 3 characters are there. So what will be the formula for gametes? What will be the formula for phenotype? And what is the formula for genotype?
2 to the power n and 3 to the power n.
Yeah, it's a 2 to the power n and 3 to the power n. So 2 to the power 3 and 3 to the power 3. So 8:27. So the answer is C. So finally, what is meaning of dihybrid cross? It's a breeding experiment between the two organisms which are identical hybrids for 2 traits. So we are taking 2 different types of traits. In other words, a dihybrid cross is a cross between 2 organisms with both being heterozygous for 2 different traits. Or simply to say, so the cross between 2 different types of characters holding parents, okay, or the plants. And what is the meaning of law of independent assortment? During a dihybrid cross, crossing of 2 pair of traits, an assortment of each pair is independent of the other pair. So one character assortment is independent of the other character assortment. So that all is independent of the color of the seed. The color of the seed is independent of the shape of the seed. So one of the character is independent of the other character. So that's called as the law of independent assortment. And guys, you don't write your own definitions. These are very important. If they come in board exam, you should need to write the definition that's given in the NCERT as it is, not in your own words. So, and remember, types of gametes, type of phenotype, genotype. So it's a 2 power n for gametes and phenotype, and genotype is 3 power n, where n is number of heterozygous gene pairs, or you can remember it like number of characters. So whether for mono, it's 1 character, so you are taking 1. For dihybrid, it's a 2 characters, so you are taking 2. For multi, you can 3, 4, any. So for mono, it's a only capital Y, small y. For dihybrid, it is capital Y, small y, capital R, smaller. For 3, it can be capital Y, capital R, smaller, capital T, small t. Okay, so you can do like that. So understood? So these are the calculations present in this chapter.